Drop it. Now let's talk about vegetative adaptation. Vegetative adaptations. If you remember, as we were talking in the morning, I spoke about I said that there are modifications of reproductive and vegetative structures, which allows the organisms to thrive in adverse conditions such as desiccation, salt concentration, presence of toxins, high temperature, lack of adequate air, and many others. So, despite all this, some microbes are able to adapt to the environment that they find themselves that's adverse. So that is why we talk about the vegetative and also the reproductive adaptations in about 30 minutes. Then we'll call it quits. Then next week, somebody else will come around to continue the lectures. So vegetative adaptations. Many cells and mycelial pieces. Many cells and mycelial, mycelial, pieces, PIECS, can become dormant and yet viable for a long period of time. Many bacterial cells may form a protective capsule of polysaccharide and polypeptides. Mycelia may also become resistant by having thick ornamental walls. Or develop into compact, highly branched knots. KNOTS, highly branched knots, called sclerotia. Sclerotia. Sclerotia may survive in the soil for several years. Years, not weeks, not days, years. Many pathogenic fungi produce sclerotia as a means of temporary survival. Many pathogenic fungi produce sclerotia as a means of temporary survival in the absence of the host. Now let's talk about reproductive structures. Many soil microbes form special resistant spores. Which are usually larger and thick-walled. which are usually larger and thick-walled. That's a wall that is thick. E.g. include the following. Number 1, bacterial endospores. E.g. Bacillus and Clostridium. E.g. Bacillus. On the board is an endospore of Bacillus species. Such spores are resistant to radiation. Such spores are resistant to radiation, desiccation, many chemicals, including antibiotics. Many chemicals, including antibiotics, and high temperature. Sometimes, Bacillus endospores may survive a temperature between 80 to 100 degrees C. Sometimes, Bacillus endospores may survive a temperature between 80 to 100 degrees C for 30 minutes or more. The next one are spores of actinomycetes or blue-green algae. Spores of actinomycetes or blue-green algae. Spores of actinomycetes, Streptomyces species, and Thermoactinomyces. Thermoactinomyces vulgaris are known to be resistant to many adverse conditions. But essentially to desiccation. Some blue-green algae produce either some resting spores called akinets, A-K-I-N-E-T-S. Some resting spores called akinets, or some large vegetative cells called heterocysts. Vegetative cells called heterocysts. Let's look at fungal spores. These are under the reproductive structure: bacterial endospore, spores of actinomycetes, of living algae, and then bacterial spores now.
Fungal spores.
Fungal spores in form of conidia. Fungal spores in form of conidia, chlamydospores. Fungal spores in form of conidia, chlamydospores. Can you spell it? Sporangiospores. Sporangiospores. Zygospores. Basidiospores. Ascospores, etc. In all cases, full stop, in all cases, the spores can remain dormant. In all cases, the spores can remain dormant until conditions for germination becomes conducive for all the types of spores. The chlamydospore is the most important. The chlamydospore is the most important in relation to survival. The chlamydospore is the most important in terms of, in relation to survival, or in terms of survival. Its thick wall has a dense liquid content. Its thick wall has a dense liquid content. A limited power of growth and a long viability period. A common example is found in Fusarium or Zisporium. A common example is found in Fusarium or Zisporium. Methods of studying soil microorganisms. The first method, number one, microscopic methods. Under that, you have A, stained soil preparation. That's number one in microscopic method. Then A, stained soil preparation, like gram staining. You take the soil and then you stain the soil. You have some stain, you add to the soil, then you look at it under microscope, and you see the organism you are looking for. B, Contact slide method. Contact slide method. Then the number C is direct soil examination. Direct soil examination. Number 2, culture methods. Under the culture method, A, plate culture method, or soil dilution plate method. That is the common one that we use in the lab. Plate culture method or soil dilution plate method. Another one, which is B, under culture method, is WARCUP, workup soil plate method. Then the next one is selective culture method. Then the D is soil enrichment method. So that's that. So how do you sterilize soil? Applying it. How do you sterilize? Soil sterilization can be carried out in the microbiology laboratory. Discuss how.
The application of heat.
It's a full question to discuss how. Soil sterilization can be carried out in the microbiology laboratory. Discuss how.
Sterilization involves taking the microbes or the living organism in the substance. So to get the microbes, the living organism in the soil, through scan method, the soil is put in a metallic container and subjected to heat. So the heat will denature the enzymes in the living organisms. And at times through oxidative destruction of the living organisms.
Are you writing? He's discussing with. Are you writing this? Why are you not writing now? All what you're saying, it doesn't represent 400 level for me. You say that you use heat. Is it heat from the sunlight? You know, we talk about sunlight that time, Abi. Maybe it's that heat he's talking about. So you just take the plastic, you even say metallic container, so you cannot use plastic, Abi. If you have plastic that can be autoclaved, you should not use it. Just go and pour it away, or just leave it. Go and look for money and go and buy metallic container. Because that's what this guy is saying now. So use naked flame to sterilize soil. Is who wants to bail him out? Oh? Who wants to bail him out? You use Bunsen burner to sterilize soil. Wow. Clap for you. Do you want to answer the question? See this lady, give her the mic.
We can sterilize soil by dry it using an oven.
1 mark. And it's a 10-mark question. The question is 10 marks. She has 1 mark. That's 1 mark. We are continuing now.
So, the soil sample will be put in a conical flask or a beaker, then covered with an aluminium foil and put in the oven.
Continue now. It's still 1 mark. Everything you have said is still oven. Dry it, oven. It's still the same thing you are saying.
That's the idea, yeah.
Everybody is hiding their faces. Why are you hiding your faces? You don't want me to call you. Okay, let's give it to that. Where's that top guy that I used to ask to sit down here? What? What? While you lie. Ah. They sent for him. Give it to the person that said they sent for him. So if you are giving a soil now, you have to sterilize the soil. What are you going to do now? Because you are saying that we can find some organisms in the soil. Now we don't want organisms in the soil again. Because you want to do an experiment in the lab, you must have a control. You say that there's bacteria in the soil. You must also be able to sterilize so that there will be no bacteria in that one. And you also check. So that one will help to serve as control. After sterilizing, there should be no organism there again. So that one will now serve as your control. There are many tests that you want to do. You need to sterilize the soil. You need to remove organism from the soil. Let's take, for instance, that you want to, they say that actinomycetes or some things, they produce antibiotics. And you want to isolate the antibiotics. Why are you giving me the mic? And you want to isolate the antibiotics. Okay, bring it. You have forgotten that. So I was saying that in some cases you need to sterilize the soil when you are in the soil microbiology or doing an experiment. Now we said that actinomycetes produce antibiotics, and then your project or there's a project on antibiotics production, and you want to produce the antibiotic because normally actinomycetes will live in the soil. So how then do you get antibiotics produced from the soil and not just from in test tube? So you get the soil, you need to sterilize the soil so that all organisms will off. Then you get your antibiotics, which you have isolated previously. So from test tube, you inject into the soil. Some of the conditions we said are suitable for the growth of actinomycetes, you make them available. Because I've already sterilized the soil, so only actinomycetes that you put in the soil will be in the soil. So as it's growing and enjoying the soil, because you have made the conditions suitable, which we have spoken about, then what will it be doing? It will be producing what? Antibiotics in the soil. But this time, there's no bacteria to kill. So after a while, you can now look for a way to get the antibiotics out and use it for other things you want to use it for. Purify it or do some other things. So now then, how do you then sterilize the soil? The first thing, you have to tell us what sterilization is. You are looking at me. You didn't tell us what sterilization is. That's why you miss it. Yes, you don't know what sterilization is. Because you are saying you are going to use what? Bunsen burner. You said what? So you start defining sterilization. Because even from the material that I'm looking at, there are wrong definitions there. There is no partial sterilization. Sterilization is an absolute term. Absolute. It's not partial or no partial. In some material, they say there's partial sterilization that you remove only the pathogens. That is not sterilization, that is disinfection. So there's no partial sterilization, it's an absolute 10. According to Paul Singleton, 2007. It's a textbook I used to use those. 2007, no, 1990 something. The textbook I used to use then. Somebody borrowed it and refused to return it, but no problem. The content are still with us. The content of the book, we've already taken it so they can hold the book. That was actually my seminar topic those days. Sterilization, disinfection, preservation. And it's in the soil sterilization is there. So who wants to tell us how to sterilize soil? Number one, you define sterilization. Then number two, tell us the types of the different ways of sterilization. You can use moist heat, you can use dry heat. If you're using moist heat, how do you do it? If you're using dry heat, how do you do it? That's how you gather points. You write on this, write on this. When you say discuss, explain. You're supposed to keep talking. Now just talk one thing you are looking. Is there only one way to prepare chin-chin? Is there one way to prepare chin-chin? So you can see. Can you prepare chin-chin by using the oven? And you can also prepare by frying. Can you see now? You can prepare chin-chin using oven. You can prepare chin-chin by frying. So the same thing in sterilization. Chin-chin is made from flour. Is it the same way you mix for buns that you mix for chin-chin? No. Chin-chin is more laborious. You have to cut it. You have to spread it and cut it. You roll it, yes. So you can see all those. So you have to talk about everything. Not that I just say sterilization. It's just you use dry heat, then you now use a Bunsen burner. You know that you don't even know what you're saying. So in sterilizing the soil, you can use moist heat. When you use moist heat, what instrument are you using? Autoclave. So you know all these things, but you don't want to say them. Then dry heat, what do you use? Hot air oven. Is there cold air oven? What temperature is it in the oven? And what time? 160 to 170 for 1 to 2 hours in the oven. So usually we use the maximum. You don't use the minimum. You don't use 1 hour, use 2 hours so that you are sure. Then in the autoclave, pressure. What is the pressure? What is the standard unit for pressure? What?
Pascal.
This guy is say, are you shy or what? I don't understand.
Pascal.
But if you are with your friends now, you start shouting. For you to talk in the class now, you can't talk. So the SI unit for pressure is Pascal. So why are you telling me PSI? So in Pascal, what is the pressure? You still have to go and study. Then what is the time? 15 minutes. So the moment I put on the autoclave like this, 15 minutes after, I should this autoclave. I like to do what? To cool. So all those things, you have to know them. So by next week, somebody else will be around. It's extra now. All those last ones, extra. So